Welcome to the Deep Dive. Today we're tackling a really fundamental puzzle in cell biology, how the master growth switch, mTORC1, actually gets turned on. Because for years, there's been this paradox.
That's right. We know mTORC1 controls cell growth, and we know its key activator is this protein, RHEB. But the weird thing was, in lab setups, you needed, frankly, ridiculous amounts of RHEB, way over 100 micromolar, just to get mTORC1 to show any activity.
Yeah.
That's not what happens in a cell.
Right, totally unphysiological. So what was missing? Well, the sources we're looking at today pinpoint the lysosomal membrane itself. It's not just a location, it's like a mandatory piece of the machinery.
Exactly, a structural cofactor.
Yeah.
And we're diving into this really elegant cryo-EM paper that maps out how this membrane orchestration works, step by step. It basically defines the cell's critical and gate for growth.
Okay, so before we jump into the structures, just a quick refresher. mTORC1 needs two signals to fire, right?
Correct. First, it needs to know nutrients are available. That signal comes through the RBAG GT paces and this complex called Ragulator. Think of Ragulator as the initial docking clamp. It pulls mTORC1 onto the lysosome surface.
Got it. Nutrient signal docks it, and the second signal...
Growth factors. That signal is relayed by RHEB, specifically RHEB loaded with GTP, and conveniently RHEB is also tethered right there on the lysosome surface.
Okay, so RAGs bring it close, RHEB is waiting nearby. That's the A and D gate concept. Both signals have to be true at the lysosome. But proximity alone didn't explain that massive activity boost needed.
Precisely. So the researchers in Figure 1 asked a direct question. If we rebuild this whole system on a membrane, can we get activation with normal physiological amounts of RHEB?
So they had to mimic the lysosome in a tube. How'd they do that?
They use something called LUVs, large unilamellar vesicles, basically lab-made lipid bubbles that act like a membrane surface. And they put tethered RHEB and the Rag-Regulator complex on these LUVs at concentrations much closer to what you'd find in a cell, like 250 to 300 nanomolar.
Okay, so realistic conditions, finally. What happened?
Get this. When all three were present, the LUVs, the RHEB, the Rag-Regulator, mTORC1 activity shot up. We're talking more than 35-fold higher than just mTORC1 floating around alone.
Wow, 35-fold. Okay, so that really nails it. The membrane context is non-negotiable. It solves the concentration problem.
It absolutely does.
But wait, if the membrane is acting as a scaffold, does its actual shape matter? I mean, lysosomes aren't perfect spheres, and mTORC1 is a pretty chunky complex.
That's a great thinking question. If you're trying to park this big dimeric protein machine onto a surface, does the curvature of that surface make a difference?
So did they test that?
They did in Figure 3. They compared activity on larger, flatter LUVs versus smaller, much more curved ones. And it turns out geometry is critical. On the small, highly curved liposomes around 67 to 81 nanometers in diameter, the mTORC1 activity dropped by about 65% compared to the larger, flatter ones.
65% reduction. So the complex literally needs enough flat real estate on the lysosome to sit properly and work at full tilt. That's fascinating.
It really is. And this leads right into the structural details they uncovered with cryo-EM, shown in Figure 5. It's not just one step, it's a sequence.
Okay, break it down for us. What are the steps?
Right, so they identified a clear four-step activation pathway. Step 1 is recruitment. Rag-Regulator brings mTORC1 close, maybe within about 100 angstroms of the membrane.
Step 1, get in the vicinity.
Exactly. Step 2 is capture. RHEB GTP grabs mTORC1, pulls it even closer, like within 40 angstroms, and this capture triggers a big conformational change in mTORC1 itself.
Okay, so RHEB reels it in and starts to reshape it, but it's still not fully on, right?
Right.
When does the membrane itself get physically involved?
That's the cool part. Steps 3 and 4. In Step 3, a part of the mTORC1 complex called RAPTOR makes a first direct contact with the lipid membrane. It uses these two specific hydrophobic residues, like a little finger, they call it the FM finger, to touch the membrane. This gives you intermediate activation.
Intermediate activation, so not full power yet. What's the final step?
Step 4 is the final docking. The actual mTOR kinase subunit itself reaches down and contacts the membrane using a loop rich in basic positively charged residues. This final contact seems to lock everything in place, properly orienting the active site. And that achieves full catalytic activation.
So recruitment, capture, RAPTOR, touch, mTOR dock. It's like a combination lock where the membrane interaction is the final click.
Precisely. The membrane isn't just passive, it's actively participating in the final alignment.
This changes things, doesn't it? Especially for diseases like cancer, where mTORC1 is often stuck in the on position. What are the implications for therapy?
It opens up totally new therapeutic angles. For years, people focused on inhibiting the kinase active site directly, but that can be tricky, sometimes hitting other related kinases. Now you could potentially design drugs that don't block the active site itself, but instead block those crucial membrane contact points like the RAPTOR FM finger or the mTOR basic loop.
Ah, so you'd prevent the final docking steps needed for full activation. Allosteric inhibition, basically.
Exactly, it could be much more specific.
That's really promising. Okay, let's recap the 3 big takeaways for everyone listening. First,
First, the lysosomal membrane is an active player. It acts as a scaffold that solves the RHEB concentration paradox by making it potent at normal levels.
second, full activation isn't just RHEB binding. It requires direct, physical, and sequential contact with the membrane, first by RAPTOR, then by mTOR itself.
And third, the physical shape, the curvature of that membrane platform actually matters. It tunes the enzyme's activity rate.
Okay, great summary. So one last thought to leave everyone with.
Well, we know lysosomes aren't static. They change shape, they form tubules, they swell up, especially under stress. So could the cell be intentionally changing lysosome membrane curvature as another way to quickly dial mTORC1 activity up or down? It adds another potential layer of dynamic control to think about.
A very intriguing possibility. Definitely something to mull over. Thanks for walking us through that complex process.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.